So welcome to the continuation of the Bacteriology Spot. So we start. Question 11. Above is an endoscopy of the large intestines. Identify the bacteria that causes such a presentation shown above. Then choice B, what are the major toxins produced by the bacteria causing the disease shown above? What is the primary mode of transmission of the bacteria shown above? What are the hallmark symptoms associated with the bacteria? So what is this presentation before we know the bacteria? Answers on the chat, or you can unmute. What I can see are yellow plaques. So this is the large intestine, and then these are yellow plaques, outpoachings out from the large intestines. So what presentation is this? Who knows? Pseudomembranous colitis. Very good. So this is pseudomembranous colitis. Okay. Now, the bacteria responsible for this is Clostridium difficile. Okay. So this is very specific, and only pseudomembranous colitis, only Clostridium difficile is associated with this. So what happens is it can be due to the death of normal flora in the large intestines due to maybe overuse of antibiotics, for example, clindamycin, ampicillin, and cephalosporins. Okay, they kill the normal flora of the large intestines, leading to the overgrowth of Clostridium difficile. Now, Clostridium difficile has a toxin that is going to cause such a presentation. Okay, so the bacteria is Clostridium difficile. The toxins are toxin A and B, enterotoxin and cytotoxin. Okay, these toxins damage the enterocytes, the cells that line the large intestines. Then primary mode of transmission is fecal-oral. Hallmark symptoms. So this is a GI disease. So any GI symptom that you have, you just mention it. So watery diarrhea, abdominal pain, fever, and leukocytosis. Leukocytosis means an increase in the level of the white blood cells. Okay. So remember, I told you any bacteria that you're asked and you're asked on symptoms, just remember fever. Fever must be in all the bacteria. Okay. Now, question 12. The above image is a culture from a patient's urine sample. Identify the bacteria shown. Which agars can be used to culture the bacteria shown? What hospital-acquired infections are associated with the bacteria? And which test can be used to confirm that the bacteria shown is encapsulated? So we did choice D. Choice D, the answer is Quelang reaction, or the Quelang test. What about the rest? What bacteria is this? So I give you a hint. There's only one bacteria that when you grow, it will give you a mucoid appearance on the plate. Okay, there's this mucoid appearance. So what bacteria are we thinking that has a mucoid appearance?
Proteus.
Proteus, no. Klebsiella, very good. Klebsiella. Okay. So which agars are used to grow Klebsiella? Considering it's a lactose fermenter, which one do we use? How do we grow our lactose fermenters? McConkey, good. So you can use McConkey, you can also use Cled medium, very good. Which hospital acquired infections associated with the above bacteria? So try and think hospital-acquired infections. So I can give you a hint. Number one, pneumonia. Pneumonia is very common. Which other one can be hospital-acquired? UTI is very good. Okay. So this is Klebsiella due to that mucoid appearance, McConkey and clade. So remember on McConkey, lactose fermenters will turn pink. You'll see pink colonies on McConkey. Then we have pneumonia, UTIs, bloodstream infections. So it's sepsis and wound infections. Then the test is the Quelang reaction or capsular swelling test. Okay, this was direct. So if you just knew that there's this mucoid appearance, you know it's Klebsiella, you can answer all these other questions. Okay, this is another one. The above is a petri dish from a bacteria cultured on McConkey. Okay. Now, this is definitely a lactose fermenting bacteria. So they'll ask, what is the primary use of McConkey agar? What are the functions of bile salts and crystal violet as components in the McConkey? Then outline 4 lactose fermenting bacteria. Okay, so why do we use McConkey? It has only one function. So the function of McConkey is to show you the difference between lactose fermenting bacteria and non-lactose fermenting bacteria. Okay, so it's the differentiation. Bile salts and crystal violet, they are components added in McConkey. Their function is to inhibit the growth of gram-positive bacteria. Okay, because we want only gram-negative bacteria to grow, we use the component of McConkey as bile salts and crystal violet to inhibit that growth. Then 4 lactose fermenting bacteria. This is direct. The mnemonics usually 6. Somebody type on the chat. Or you can unmute. Citrobacter, Enterobacter, E. coli, and Klebsiella. Very good. So McConkey agar is primarily used for isolation and differentiation of gram-negative enteric bacteria, particularly for distinguishing lactose fermenters from non-lactose fermenters. So the use is the differentiation, the aspect that it is able to differentiate. Then bile salts, it inhibits growth of gram-positive bacteria, just allowing gram-negative. So we have Citrobacter, the lactose fermenting, E. coli, Enterobacter, Klebsiella, and Serratia. Okay. This was a direct question. We go to next. Identify the shape of the bacteria. So this one, you just had to comment, okay? 3 gram-negative bacteria that take up the above shape. Then apart from the shape, mention 3 other shapes bacteria can take up. So if you were to do a workup, so answers on the chat. If you were to do a workup, remember how we classify gram-negative bacteria? There is the diplococci, there is the bacilli, there is the cocobacilli, and then there's this one, which is curved, okay? Comma-shaped, or you can say curved, okay? Then what are the 3 gram-negative bacteria that take up this comma shape? We have Vibrio cholerae, good. We have Campylobacter jejuni and Helicobacter. Okay. Then the other shapes I think I've mentioned when we were classifying this. So curved, you can use curved, you can say comma, Vibrio, Campylobacter, and Helicobacter. And then we have cocci. So cocci, we have the diplococci, which were the Neisseria gonorrhea, and Neisseria meningitidis. Then for the bacillus, we had the lactose fermenting ones, okay? E. coli, Klebsiella, Enterobacter, Citrobacter, and Serratia. And then the non-lactose fermenters, we had Pseudomonas, Salmonella, Shigella, Yersinia. They are all bacilli. Then spiral. So spiral are things like, Treponema pallidum, those ones, okay? The one that causes syphilis. We have Treponema pallidum, we have Leptospira, and this other one, Borrelia, okay? Those are the spiral-shaped bacteria. Yes, if you wrote diplococci, still part of the cocci, but that can also be an answer. Then we have the coccobacilli, the ones that are they're spherical and rod-shaped, Haemophilus influenzae, and Bordetella pertussis. Okay. So, if you get the principle of how these bacteria are shaped, it's you're easily able to classify them. Then we go to the next one. Identify the above presentation. So you can see here there's some swelling on the inguinal region of this patient. Then what's the causative microorganism? Then the animals that are reservoirs for this organism. Then 3 main forms of the disease caused by the bacteria. Okay, so what is this presentation? Buboes, very good, buboes. What causes buboes? Yersinia pestis, very good. So please don't confuse Yersinia pestis and Yersinia enterolytica. Yersinia enterolytica is purely a GI bacteria. Okay, it causes bloody diarrhea. Okay, that's Yersinia enterolytica, but Yersinia pestis causes buboes. Then the reservoir. So these buboes are the ones that cause the plagues. Okay. What is the reservoir for Yersinia pestis? It's mainly rats, okay? The rodents, good. Yes, so I've seen someone has said flea. Flea is how it is transmitted, okay? So it is transmitted mainly by flea bites, but the reservoir is the rodents, okay? If you can remember Yersinia pestis, maybe something to mention is that it has the bipolar staining, the safety pin appearance, okay? And it mainly causes, so the 3 forms are the 3 forms of the plague. So we have bubonic plague, septicemic plague, and pneumonic plague. Those are the 3 forms of the disease caused by Yersinia pestis. Okay, please don't confuse this with Yersinia enterocolitica. Okay, number 16. The above image demonstrates extensive skin peeling in a child. So you can see The peeling has started from the forehead as it extends to the chin. Okay. Identify the condition. What is the most likely causative organism? What toxin is responsible for causing the above condition? Then which lab tests can be used to confirm toxin produced by the organism causing this condition? So you have to know, why is this child peeling? So which organism can you think that causes skin infection, especially this peeling-like infection? So identify the condition of the bacteria and then the bacteria responsible for this. So this is Staphylococcus aureus, scalded skin. So it's usually called SSSS, staphylococcal scalded skin syndrome. Okay. I've seen someone has mentioned strep pyogenes. So please, let's not. So strep pyogenes, yes, it can cause a skin infection, but mostly impetigo. Okay. If you see peeling in a, in a child or in a patient, that is Staphylococcus Staphylococcus scalded skin syndrome. Okay. For Streptococcus pyogenes, it mainly causes impetigo. Okay. How you'll know the difference is impetigo is we refer it to as honey crusted lesions. Okay, they're yellowish, goldenish color and appearance, and they're not like, you won't see this exfoliative appearance. It's mainly like a lesion here, a lesion here, a lesion here. Okay, that's for Streptococcus pyogenes. Okay, so please don't confuse those two. This is Staphylococcus scalded skin syndrome. Okay, so which toxin is responsible for SSSS? Exfoliative toxin. Very good. Okay, don't confuse. You see, these toxins, it's good so that you know what they do. If you can remember, when we did Staphylococcus aureus, there were types of toxins we mentioned. There is the exfoliative toxin. Okay, there is the protein A. Protein A, we said it binds to the Fc portion of IgG, inhibiting complement activation. Then there is the toxic shock syndrome. You see, toxic shock syndrome, this is a super antigen. Okay. Then we have this scalded skin syndrome. Then we have the one for food poisoning, which is an enterotoxin. So it's very good you know these toxins, because you can be asked. For example, they can bring in an MCQ and they tell you which one is incorrectly labeled. So they say maybe toxic shock syndrome. Okay, so don't confuse. There's toxic shock syndrome and there's toxic shock-like syndrome. Okay, for toxic shock-like syndrome, it's caused by Streptococcus pyogenes. It is similar to that. But for Staphylococcus is toxic shock syndrome, which is a super antigen. Okay, then the lab tests. So lab tests for toxins, we mainly use ELISA. So let's see the answers. So Staphylococcus scalded skin syndrome, Staphylococcus aureus, exfoliative toxin. We use PCR for the genes and ELISA for toxic detection. Okay. I hope this is clear. Then we go to this. So this is a very, very high yield image because you must know this. Okay. Identify the above presentation and the bacteria responsible for it. Mention the disease caused by the bacteria and its primary vector. Identify the 3 stages in pathogenesis of the disease, mentioning 1 symptom in each. What is the drug of choice for this disease? So remember, when we were talking about the zoonotic diseases, Can you remember? What did we say? Because before we even get into this, what did we say about the zoonotic diseases, the zoonotic bacteria? What drug of choice is used? We mentioned that you must not forget any zoonotic diseases. Zoonotic diseases are quite a few. What did we say?
Penicillin.
Very good. Penicillin. Did you say penicillin? No, we didn't say penicillin. We said doxycycline. We use a tetracycline for all the zoonotic diseases. Now, this is called target rash. Okay. As you can see, it looks like a target. You can also call it bullseye appearance. Okay. What bacteria is responsible for this? What bacteria is responsible? Borrelia. Okay. Borrelia burgdorferi. Okay. Mention the disease caused by the bacteria and its primary vector. So this is Lyme disease. The primary vector is the tick. Okay. Now let's see. Target rash in Borrelia burgdorferi. You can also say bullseye rash or bullseye appearance rash. Okay. Then Lyme disease, ixoides tick. You can just mention tick. Then for the stages, if you can remember, there were different stages in appearance of Lyme disease. Stage 1, it was localized, okay? So appearance of this rash, and then you're going to have flu-like symptoms. So flu-like symptoms are things like cough, blocked nose, malaise, headaches, runny nose, okay? Things like that. Then stage 2, this is when dissemination occurs. So this is early dissemination. You can have carditis. Carditis means inflammation of the heart muscles. We have Bell's palsy. Bell's palsy is when you have, it has affected the facial nerve. So one side of the face is going to be affected for facial nerve palsy, which is Bell's palsy. So you're going to have drooping on that side, myalgia, which is muscle pain, and then arthritis, which is joint pain. Then for stage 3, this is now the late dissemination. Late dissemination stage is when now it has affected multiple organs. Encephalopathy, this is when the bacteria reaches the brain. Peripheral neuropathy, when it affects the nerves. And then chronic arthritis, when it affects the joints. Okay. So once you see this target rash, you have to give the patient doxycycline. So the good thing is this is not common in Kenya, but it's also a high-yield thing you should know. Lyme disease, there's actually a place in the US known as Lyme. So that's why it was mentioned, it was named after that. It is on the areas of northeastern USA. Okay. But this is very, very high yield because this is the only bacteria that causes such an appearance. Then for drug of choice, we said doxycycline. Then remember, doxycycline is not recommended in pregnant women. It's a teratogen, and it is not recommended in children because it would cause decolorization of their teeth. So instead, we give amoxicillin. Then ceftriaxone is used for stage 2 and stage 3 of Lyme disease. Okay, stage 1, we use doxycycline. Stage 2 and 3, we give ceftriaxone. IV therapy, so intravenous therapy. Number 18. So I think I had mentioned this. So what appearance is this before we identify the two bacteria? What is odd about this picture? What can you see? These are rashes, okay? There are rashes on the palms and the sole of the feet. You see, most of the time, rashes usually spare the palms and the soles of the feet. But in this case, it was affected. So there are only 3 microorganisms that cause rashes to appear on the palms and the soles of the feet. And 2 of them are bacteria. So which bacteria are these? There's one that everyone must know. Which one?
Could that be treponema?
Yes, Treponema pallidum. And then the next one, maybe you couldn't know, it's Rickettsia rickettsii. Okay. So syphilis and Rickettsia rickettsii is causes Rocky Mountain spotted fever. Okay. So syphilis and Rocky Mountain spotted fever. The other one, who can try? Who knows the virus that causes this appearance? Who wants to try? Who wants to try? It's a virus. Measles, no, measles spares the palms and the soles. Hand, foot, and mouth disease. Very good. So the virus is called Coxsackie-A virus. Okay. Do you remember Coxsackie? It was one of the picoRNA viruses, together with polio, rhinoviruses, hepatitis A, and yeah, those ones. It was picoRNA. Okay. So in case you were brought this and you asked the virus, the answer is Coxsackie-A, or hand, foot, and mouth disease. Okay. This is, what organism is this? Is this a flea? Is this a tick? Is this what else? Is it a louse? Is it a mouse? No, not mouse. Mite? Is it mite? So what is this? Because you have to know what this is before you answer the bacteria that are transmitted by this.
I think it looks like a flea.
Yes, it's a flea. So what is transmitted by a flea bite? We have done one.
Yersinia
pestis. Yes. Another one?
Rickettsia typhi.
Rickettsia typhi. Is there another one? I think those are the main ones. Rickettsia typhi and rickettsia. There's also this rickettsia felis. Yeah. You have a question?
No, I was just saying it's Rickettsia and Yersinia.
Yeah, those are the common ones. I'm seeing someone has said Shigella. Oh no, Shigella is not a flea-borne. How is Shigella spread? In fact, Shigella is not a zoonotic disease. It's only confined in human beings. How is Shigella spread? Considering it causes diarrhea. Contaminated water, yes. Fecal oral. Okay. Now, this one we did it. So you should be telling me the answers. This is a triple sugar ion, partial fermentation with H2S production. But it reads through flies. This is not a fly, this is a flea, not a fly. House flies are different from fleas. This one has no wings, it moves by jumping. Flies, okay, those are different. Name at least 4 common bacteria that produce H2S on TSI agar, similar to this. First of all, who will tell me what TSI is this? Tell me, so you tell me the bat, no, so you tell me the slant, the bat, gas, and H2S. We did this yesterday. So what is the anyone wants to try? What is this TSI? Alkaline slant. So definitely it's alkaline slant. Then question mark on the bat, gas, and H2S. Okay. So with gas, yes, gas is also there. You can see that from this bubble. Okay. Now, as we observed yesterday, if the bacteria is a fermenter, both lactose, glucose, and sucrose, what will be the color of the bat? Yellow, good. So it's acid. It's just that we can't see, but keep in mind that, okay? But on exam, definitely they can't bring for you this and then they tell you describe the TSI. But I just want you to keep in mind that beneath this H2S production, there's an acid bat, okay? So this TSI would be alkaline slant, acid bat, gas, H2S. Now, what are the bacteria that produce H2S? Remember, yesterday, there were 2 types of salmonella. Which salmonella did we say produces H2S? Is it typhi or typhimurium? Mm-hmm. You remember? Okay. So remember, both salmonella species produce H2S, but typhi produces some little H2S. Salmonella typhimurium. So if this is most likely Salmonella typhimurium rather than Salmonella typhi. Then we have Proteus mirabilis, Proteus vulgaris, we have Citrobacter, okay? And then there's this one, Edwardsiella tarda. This is one of the Enterobacteriaceae, but these are the most important. So if you write Salmonella typhi, Salmonella typhimurium, Proteus, and Citrobacter, you'll be okay. Okay. I want you guys to remember. And you see, for all of these, they are similar. Alkaline slat, acid bat, gas, H2S. Okay. Question 21. Define the term obligate aerobe. Then give 3 examples of obligate aerobes. So these terms are very, very important. Facultative aerobes, obligate aerobes, obligate anaerobes. Okay. There are terms we use. Then there's also microaerophilic bacteria. So anytime you see the word obligate, it means must. If I tell you you're obligated to do something, it is mandatory. It is a must to do something, okay? So obligate aerobe means that it must grow in the presence of oxygen. If there's no oxygen, it will not grow. If there's oxygen, it will grow. Okay. Obligate anaerobe. It grows in the absence of oxygen. It requires no oxygen for growth. Facultative, this means that it may or may not require oxygen. It can use oxygen, but it can switch to so you know the presence, why we need oxygen is because we require oxygen to produce ATP. Okay, so facultative anaerobes can use oxygen to produce ATP, but they may switch to other methods to produce energy. For example, anaerobic metabolism, where fermentation takes place for it to get its energy. Okay. Now, microaerophile or microaerophilic bacteria or microaerophile are that grow in subatmospheric oxygen levels, for example, in water. Okay. Now, examples of obligate aerobes, these ones you should be knowing. Think of bacteria that affect the respiratory system. Answers?
Mycobacterium tuberculosis.
TB, very good. Pseudomonas, good. Nocardia, yeah, things like that. Okay, there are quite a few. So TB, Pseudomonas, Bacillus, Nocardia, Micrococcus, Bordetella, Brucella, Neisseria, and both Neisserias. Okay, those are obligate aerobes. Now, look at this. So I think everyone got this question. Name this selective media, type of bacteria the above media is used to culture, characteristic motility of the above bacteria, primary mode of transmission, and then Gram-Stein morphology of the bacteria. I think everyone got this. So what is this? What is this? Who knows? Who knows this? Who knows this? Okay, let's go through it. So this is Skyros agar, used for Campylobacter jejuni. You can see there are white colonies on a brown background. Okay. Motility, I feel like motility should have given it away. We have the corkscrew motility. It moves like a corkscrew, going round, 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 round. Then primary mode of transmission for Campylobacter, it is mainly fecal-oral. So consumption of contaminated food, undercooked poultry, unpasteurized milk, and contaminated water. And then what is the most likely gram stain morphology? We have it is gram negative, curved. Remember we said the curved are Campylobacter, Helicobacter, and Vibrio. S-shaped or spiral rods. Okay. Then we have this. The image provided shows a urease test result, which color changes indicate a positive urease test. Then name 2 urease positive organisms, bacteria that can be identified using this test. So I think here's where we were given the mnemonic. Bernadette, did you give us a mnemonic for this?
Yes, I did.
It was P, what something punch?
P chunks.
Chunks, yeah, not punch, chunks. Yes, so for positive, it's usually pink, then yellow means it's negative. So you said P chunks, I'm going to add here. So P was? Let me even edit it. You said, let me add those other species. So Proteus, can you read it for me?
P was Proteus. The E, the 2 E's are not relevant.
Okay,
C
yes.
was Cryptococcus.
Oh, but here they've asked bacteria. So crypto, remember it's a fungus.
Oh, yeah. Yeah. H, Helicobacter. U for Ureaplasma. N for Nocardia. K for Klebsiella. S for Saprophyticus, and the other S for Epidermidis.
Or Staphylococcus.
Yeah.
Epidermidis and Saprophyticus. Okay. Remember, Saprophyticus was the one that causes UTIs in sexually active women. Okay. So I've added the rest. You can add here, Cryptococcus neoformans. It's a fungal infection. Okay, thank you. Then you can see these are diplococci or kidney-shaped bacteria, which is very specific for a type of bacteria. And you can see from here that they're gram negative. Okay, so that will rule out strep. If you're thinking strep, no, these are gram negative. Then you can see, name 2 pathogenic species associated with this bacteria. What are the main virulence factors of any of the 2? Then a lab technician isolated gram negative Diplococcus that is oxidase positive, ferments glucose, but not maltose. What is the most likely pathogen? So this is a very common question. So I became so direct, but if you can remember, we said Neisseria, gonorrhea, only ferments glucose, and then Neisseria, meningitidis ferments, both glucose and maltose, okay? Then for the virulence factors, so Neisseria gonorrhea and Neisseria meningitis, we have pili, IgA protease, outer membrane proteins, lipooligosaccharide in Neisseria gonorrhea, then lipopolysaccharide in Neisseria meningitidis. There is the capsule, endotoxin, which is the LPS, IgA protease, and pili. Where else did we find IgA protease? You guys remember?
Streptococcus pneumoniae.
Very good. Very good. And Haemophilus, yes. Then the last question, discuss the treponemal serological tests. So if you can remember, there were treponemal and non-treponemal serological tests. Did anyone get this question? Did you do the research? For syphilis, the treponemal and non-treponemal tests. What that means is non-treponemal tests means that it is non-specific, meaning that other bacteria or other diseases can give you a false positive for the non-treponemal test. But treponemal, they are very specific for treponema. So who wants to attempt? What are the treponemal? And remember, these are serological tests. So there's no PCR, dark field microscopy. Those are not serological tests. Okay, all right. So non-treponemal, we have VDR, can test, rapid plasma reagent. So these ones, for example, like VDRL, can give you a false positive. If you have things like lupus, if you're pregnant, some drugs can also give you a false VDRL test, and some other viral infections, for example, Epstein-Barr virus, okay, and hepatitis, they can give you a false VDRL positive. And then treponemal, these are now the specific ones. We have treponema pallidum hemagglutinin assay, TPHA, fluorescent treponemal antibody absorption assay, IgM, FTA ABS test, used for diagnosis of congenital syphilis. We have ELISA, and then treponema pallidum immobilization test. Okay, so those are the treponemal tests, very specific for treponema. So the others, I saw many of you were writing dark field microscopy. Dark field microscopy is not a serological test. It is a direct testing method. So what happens is we don't usually rush to these tests for a patient we think they have syphilis. We start with VDRL. Then if VDRL is positive, we now do this one, FTA ABS, fluorescent treponemal antibody absorption assay, to check now if they're truly positive, because you can have false positive, tests. These VDRL and rapid plasma reagent, they're known as screening tests. Okay. Then these ones are definitive tests. So this question would have been asked, discuss the definitive serological tests of treponema. You answer this. If they ask, discuss or mention 2 screening tests for treponema pallidum or syphilis, it's VDRL, can test, and rapid plasma reagent. Okay. Is that okay?
I have a question.
Yes, yes.
Okay, so now the question is saying discuss, but like we're just listing the names of the tests. Like if we get this question, do you have to like state specifically what like antigens they're looking for, or?
Yeah, ideally, if they say discuss, that's what you're supposed to be doing, because here I've listed. In fact, let me even edit that. So this should be in brackets. This is not an answer. Okay. This is in brackets. These are non-treponemal, then these are treponemal. Yeah, so ideally you should be doing that if they say discuss. Also, the marks should guide you. If you see discuss and it's 2 marks, just mention. But most likely they'll tell you to list rather than to discuss. Discuss is more of an essay question. Okay, but if it's spot, they'll tell you that. Okay.
Okay, thank you.
Okay. Yeah, so I'm done. Any question?